Hey everybody, welcome to a video of Alstein. So in this video, we're going to be talking about transcription. Now you might be thinking, hey Albert, why are you sharing a screen? That's because today we're going to be using a simulation. So I'll put the link to the simulation in the comments below. So if we go here, you can see that there are these weird bulb-like thingies. And they're just hanging around, sometimes on the DNA, sometimes just over here. And you can see there's a regulatory region and there's a transcribed region. Now, here we have a few sliders. We have a positive transcription factor, concentration, and affinity. And we also have our RNA polymerase affinity. So, basically, the positive transcription factor just allows transcription to happen. So as you can see, one of the positive transcription factors has bound down and there it goes. As you can see, there's another one. The RNA polymerases are binding onto the transcribed region here. They're changing shape and they're producing these mRNAs that are then released at the end. So you can see if we have a negative transcription factor, it actually blocks, if we remove this, it can block the amount, it can block transcription, so that the positive transcription factors can bind. For example, if we turn the concentration to none, then it can bind. So let's go down here to the expression. Now we're not going to care about these ribosome thingies or mRNA destroyer things. Well, technically we can add these two in. And if we see here, there are, we can put the positive transcription factor onto the regulatory region, and then eventually the RNA polymerase will come here and it will start transcribing. And they will and you might notice that there's this protein collection thingy, and that's for the ribosomes, but the ribosomes we're not going to care about today. So now that we've seen how this kind of thing works, let's see what it actually, the technical stuff and how it works and how that thing works. Okay, so now that I've drawn this strand of DNA, you might have noticed that I unwound it, kind of. Yeah, and that's exactly what I did. So, what the DNA polymerase is going to do is it's going to take these, and it's going to flip them around. Not precisely like this, but, yeah, you get the idea. They unzip it and then they take some of the bases away from the other, so they're flipped down like this. And I don't think I need these or that. Anyways, and then some free bases from the outside, kind of they come to them and they attach. So this one there. I guess I'll draw them. And then another one gets attached, and then another, and another, and another, and another. And suppose this is the transcript, the area that needs to be transcribed. So then these bases get attached, and the sugar phosphate backbone, which is something that you can research, I'm not going to be talking about it, but the sugar phosphate backbone forms around these bases and turns them into a strand of mRNA. Like we saw in the simulation, it's the thing that floated away. Now, usually, this is not as simple. Usually there are millions of bases in one transcribed region. And the DNA polymerase actually works really, really quickly. And basically, then what happens is the RNA goes through a kind of cleansing thingy. So So let's say, actually, let's just zoom in a bit. So say this is A, this is U, remember this is mRNA, this is RNA, not and then this is T, if this is T, if this is A, and then a G, and C, and so on, dot, dot, dot. And now, let's say that there's like A. Now, let's say that this is the front of the mRNA, and this is the back of the mRNA. The mRNA is then going to go to a complex, and this complex is going to add an AUG to the front. Actually, you know what? Why not let's just let this side be the front and this side be the end. So it's just going to add an AUG here. And by the way, if you don't know what this is this amino acid this base configuration is also known as the amino acid configuration, methionine or M. But we're gonna go over that later. So anyways, so then we have this AUG here in front, so then it appends that. It's like kind of like a worker's cap to signal that you're a worker. And then it's gonna add in like a million A's at the end. And I'm not joking when I say a million A's. It's literally A and so on. That's not typing. I'm confused. Okay. I'm confused. Wait, what? Okay, whatever. So then you get the idea. There's gonna be like a million A's stringed on the like the back, kind of like a tail. And then what's gonna happen is this thing over here, this like the modified RNA is gonna go on, and it's gonna go into a region outside of the nucleus. This is all happening inside of the nucleus. So this is all inside of the nucleus, including the DNA, of course. So that's the nucleus. Then it's going to go outside the nucleus into the cytoplasm. If you don't know what these are, you better watch the first video that I made in the biology series. And then it's going to go into translation. Now, translation is a topic for the next video, and we're not going to leave it to next week. Alright, it's time for the question of the day. So, imagine you had a DNA, and like many of my other question videos, what if it was faulty? What if it had a mutation in it? Would the mRNA carry the same mutation, or would it get fixed? Leave your answers in the comments below. Please like and subscribe, and I'll see you in the next video.